All right. So I think folks are coming in and joining, and I'll go ahead and get started with our introduction. So welcome to Charismatic Crustaceans, what Columbia River crayfish can teach us. We're super excited to have everyone here today. My name is Lori Epstein, and I am the Water Quality Director with Columbia Riverkeeper. And I'm so excited to welcome you here today. Thanks so much for joining us. We're going to be hearing from Dr. Alan Koluk from the University of Idaho, and he is going to be talking about the Crayfish Mercury Monitoring Project that's evaluating the distribution and abundance of mercury contamination across the Columbia River Basin, as well as promoting community engagement and through participatory science. I'm joining you from Hood River, Oregon, and to get things going, I love to see where all of you all are joining us from. So if you're able, you can just type into the chat where you're joining us from. One of the things I really love about these webinars is that people are able to join us and engage from all over the Columbia River Basin and even beyond. So it's really fun to see where all of you are coming from. This webinar is brought to you for free as part of Columbia Riverkeeper's Love Your Columbia webinar series. Columbia Riverkeeper is a nonprofit dedicated to the protection and restoration of the Columbia River. And we use legal advocacy and community organizing to stop pollution and fight fossil fuels and save salmon and engage communities and protect the Columbia River. And so we offer these webinars as a free way to connect and help educate people in our watershed about the different issues that are affecting the river. So personally, I'm a salmon biologist, so that might influence the fact that I find crayfish endlessly fascinating. That might kind of come to me naturally, but I do actually think that they're broadly interesting creatures. I know with my children, one of the things that they love to do when we're on river trips, kind of the first place they go is to the shoreline, looking for crayfish, trying to find them and catch them. And so there's just something about these creatures that are captivating. And I'm really excited to learn more today. I've been with Riverkeeper for about 14 years, and a lot of my time at Riverkeeper has been dedicated to understanding and mitigating the impacts of toxic pollution on the Columbia River. And mercury specifically has been identified as one of the top toxics of concern on the Columbia River. And it's one of the primary drivers for fish advisories and concerns around Columbia River fish, including salmon. And the issues with mercury are really, they're truly a global issue. Mercury can travel around the planet, in the atmosphere, and much of the mercury in the Columbia Basin comes from atmospheric deposition. But one of the things that I love about these kind of projects that we're going to be hearing about today and these community-driven participatory science is that even though we're kind of thinking about these big global issues that can feel really daunting and overwhelming, there are ways to get involved. There are ways to contribute to the science and the understanding of the impacts of these issues locally. And so as they say, if action is the antidote to despair, there's so many different ways to take action and get informed and get involved. So that's exactly what we're trying to do at Riverkeeper. And so if these are issues that feel important to you, one of the things you can do is become a member of Columbia Riverkeeper and support this work. You can do that through a one-time donation or a recurring contribution. We'll put a link in the chat, if you'd like to become a member of Columbia Riverkeeper. But a really great step is just being here today and learning about these issues. So I really want to thank you all for taking the time to join us. Before I introduce today's speakers, I'd like to start with a couple of acknowledgements. First, I'd love to start with a land acknowledgement. And we at Columbia Riverkeeper, we recognize the unique and enduring relationship that exists between Native people and their traditional territories. And we respectfully acknowledge that the places we are joining today's webinar from rest on traditional lands of Native people who've cared for these lands and waters since time immemorial and continue to do so today. I'm joining from near Columbia Riverkeeper's office in Hood River, which rests on the traditional lands of the Wasco, Wishrum, Warm Springs, and Yakima Nation. I'd also like to acknowledge the U.S. Environmental Protection Agency. who's funding this webinar through the Columbia River Basin Restoration Funding Assistance Program, which is a grants program for environmental protection and restoration throughout the Columbia River Basin. And the Crayfish Mercury Monitoring Project is also a grantee of that program. So the contents of this webinar don't necessarily reflect the views or the policies of the Environmental Protection Agency, but we really thank the EPA for the support of this webinar and of our programs. Final bit of housekeeping is that we do plan to leave time for questions at the end of the presentation. And so at any time during the webinar, you can type questions into the Q&A, and we'll try to answer as many as we can. And then we can also share contact information at the end of the webinar in case this conversation is ongoing. So use the Q&A feature for questions. And with that, I'd love to introduce today's speaker. Today, we've got Dr. Alan Kollock. He's a professor of ecotoxicology at the University of Idaho. His previous positions include being the founding director at the Nebraska Watershed Network at the University of Nebraska, directing the Center for Environmental Health and Toxicology at the University of Nebraska Medical Center, and the director of the Idaho Water Resources Research Institute at the University of Idaho. Alan's published over 95 scientific papers and has written 2 books. His nonfiction book, Modern Poisons, is a brief introduction to contemporary toxicology. And Alan very generously offered one of his books that will be raffled off today. For everyone who registered will be entered into our raffle. And his latest book project is a nonfiction assessment of chemical safety and regulation during the Second Industrial Revolution. He definitely knows his toxics, but I hope you'll find his enthusiasm for crayfish and participatory science as contagious as I do. So really excited to have him here today. And with that, I will turn it over.
Great. Lori, can you see my slides?
Yes, I can. Thanks.
Okay, great. It's tough for me to, at my age, to like George Bush said, it's tough for me to walk and chew gum at the same time. So I had to get the slides up and running before I could speak. So before I get started, I'd like to thank Lori and the Columbia River Keeper for giving me this opportunity to talk with you all today. I really appreciate it. Lori and I were talking right before this webinar began, and I was telling her how much, as I've gotten older, how much I value these community engagement presentations and talking to people about their interests in crayfish and toxic chemicals and whatever else. So again, I really appreciate the opportunity to be here. I'm going to cut right to the chase and answer the question, what Columbia River crayfish can teach us. The answer to that question is a lot. And hopefully you'll see that in the next 40 minutes or so. And then we can kind of open it up for Q&A, and I'd be happy to talk to you about whatever's on your mind, basically. So we can go from there. However, before we do that, let's talk about the problem. And the problem is this. We live, as Lori was saying when she was introducing me in that very gracious introduction, we live at the end of the Second Industrial Revolution. And the Second Industrial Revolution was really the chemical revolution. And it began in 1900, and it's continuing to this day. And as you can see from this little figure, in 1900, there were about 100,000 described organic chemicals known to science. Today, there are over 50 million. Now, in concert with that, there's also the fact that our capability as scientists to our analytic capability to measure those chemicals is going lower and lower and lower every year. So there's more chemicals, and we can understand their fate and transport in the environment at lower and lower concentrations every year. We're now down into the picogram per liter levels of concentration, as you can see from the axis on the right-hand graph. That means 10 to the minus 15 grams per liter. Absolutely unbelievable how high resolution our analytic capabilities have become. But this creates some serious problems, and we'll talk about that in a minute. Furthermore, what also happens is that we have, and you all know this anecdotally, we live in a time when there is a changing paradigm, right? We don't live in a static environment. Our environment is changing continuously. So if you think about environmental regulation and you think about the 70s, the times right around Rachel Carson, obviously DDT was a big player. When we moved into the 80s and 90s, things like PCBs and dioxins became major players. Once I got to the point where I was a scientist, back in the 90s and from 90s up until now, things like steroids and pharmaceuticals in the environment became really, really important. And now today, as many of you know, things such as bisphenol A and the latest, and we'll talk about this in a few minutes in a little bit more detail, but the latest PFAS has really taken hold of environmental regulation on a national level. So not only are there these 50 million compounds that we could potentially be interested in, but the tapestry is constantly folding, constantly changing, and our interest is constantly moving from one chemical to another, which really creates a regulatory problem. All right. This has led to individuals such as Linda Birnbaum, who is the former director of the National Institute of Environmental Health Science, to say that we are all living in a chemical soup. And the one chemical that I have on this page that I have highlighted in this little gray box, some of you may know what this chemical is. If you don't, once I say what it is, you probably will. This is 6-ppd-quinone, also known as the salmon killer, right? This is the chemical that is affiliated with particles from tires. When it gets into the water in small headwater streams where salmon are trying to reproduce, it's absolutely fatal to those salmonids. The reason that I put it on this slide is 10 years ago, 50 million chemicals. We had no idea that this chemical was doing that, right? We've created this chemical, we put it in the environment. We had no idea the devastation that this chemical could potentially cause. So given that we live in this environment with all these chemicals and our interest in these chemicals is constantly changing and chemicals are bubbling up to the surface of our interest constantly, it's daunting. It's daunting for me as an academic who's studied this for 20 years. For the lay public, whoever you are, it must be absolutely mind-boggling. So we need to think about this and we need to put it into a construct that maybe makes it a little bit easier. So I'm here to give you a little bit of Toxicology 101, and it's this. So if we're going to talk about this chemical soup, we can do two things very easily and quickly, which helps us enormously. The first thing is we can talk about solubility. We can talk about, just like if you're making salad dressing at home, right? Do the ingredients wind up in the water in the salad dressing, or do they wind up in the oil? So are things lipid soluble or are they water soluble? Because that makes a huge difference in where those chemicals go in the environment. The second thing is biotransformability. And that's just the idea that when you have a parent compound, something like 6-ppd-quinone or PFAS, does it stay in that parent form in the environment? or does it break down? Now, you all know PFAS is called the forever chemicals, right? Which kind of gives you a hint that those things aren't going to break down and they're not going to degrade into metabolites. But other things like 6-PBD-quinone do break down into metabolites. Do we need to be worried about the parent? Do we need to be worried about the metabolites? So this right here, if you remember one thing from this talk other than crayfish are really cool, and we'll get to that in a second, remember this. Water soluble, lipid soluble, break down, don't break down. That explains 80% of my advanced toxicology course right there. So you've just now, gotten a C in my toxicology 520 class. All right, so again, here's those chemicals broken down, as I just said, into biotransformable versus non-biotransformable, water soluble versus lipid soluble. We could talk about what these chemicals are in detail, and we will talk about some of them in a moment, but I just wanted to show you that you could break it down in this way, and it simplifies things a lot. All right. One of the things that Lori also mentioned before was the Columbia River Basin State of the River Report for Toxins, which came out in January 2009. And in this report, it says this, and I'm quoting the report, right? In this report, the US EPA Region 10 summarizes what we currently know about the 4 main contaminants in the basin and the risks that these chemicals pose to people, fish, and wildlife. So what are these chemicals? Well, the chemicals are all, interestingly, POPs, persistent organic pollutants. And the chemicals specifically, so in our grid, right, they fall into this one area where they're all lipid soluble, not water soluble, and they all are resistant to breakdown. That's really important. And the 4 chemicals are DDT and its metabolites. Now, DDT in the United States hasn't been used in a long time, but because it's PFAS-like in that it's for all practical purposes a forever chemical, it's still out there. And some of its metabolites are still out in the environment, even though DDT hasn't been used since the 70s. So we have DDT in the environment, we have PBDEs, these polybrominated diethyl ethers, we have PCBs, polychlorinated biphenyls, and we have mercury. And these are the 4 chemicals that are of primary interest, at least relative to the 2009 State of the River report put out by the EPA. And again, the kind of interesting thing here is they're all POPs. And that's important because when we get to crayfish, which we're going to get to momentarily, that's really important, and it actually makes our life from a monitoring perspective a heck of a lot easier. All right, the characteristics of these types of chemicals are they resist biotransformation. They're in very low concentration in the water because they're not water soluble. They're lipid soluble. So again, if you're making salad dressing, these ingredients are going to wind up in the lipid layer, not in the water layer. So very low concentration in the water, which also means that they're very hard to measure in the water because their concentrations are so low. And they're capable of biomagnification. Now, we'll talk about this in a little more detail, but just to give you a primer, biomagnification means it magnifies, the concentration magnifies as you move up the food chain. So things like phytoplankton, small single cell plants, have a low concentration. The zooplankton, the little animals that eat the plants, have a higher concentration. The little fish that eat that have a higher concentration. Crayfish have a higher concentration, and it moves up and up and up the food chain until ultimately massive Chinook salmon, or ultimately us. Okay, well, given that these things are in such low concentration in the environment, how can you measure them? If they're in parts per trillion levels in the water, how do you measure that? Well, one thing that you can do is you can use passive integrative samplers. And basically a passive integrative sampler is a system where the chemical can enter but can't leave again. So it's basically like a sponge, and the chemical gets captured and can't be released. So instead of having very low concentrations in the water, over time, this sponge will fill with chemical, making it a lot easier to analyze. So the beauty of these passive integrative samplers is that they're passive. You don't need to put any energy. You don't need batteries or anything like that. They just work through diffusion. So they're passive. The chemicals sequester into a very stable matrix, so they can't get back out. So that's great. Concentration increases over time. And this allows us to actually back calculate from that concentration in the matrix, in this stuff inside these samplers, back concentrate to concentration in the water. So it's a really nice system. And my lab group has used these for years, and they're really valuable, really useful. The only problem is they're also really visible. So if you put one out in the field and you're not careful, it's going to get vandalized. We've had that happen many times. It's really unfortunate, costs us a lot of money, really a sad thing, but that's just the reality of using these things. You got to hide them really, really well, or somebody's going to take a shotgun and blast a bunch of holes in it or do whatever, do some other nefarious thing. So given that there's this problem with these samplers in that they're, one, expensive, and two, susceptible to vandalism, is there an alternative? Is there an alternative mechanism where you can look at these POPs, these chemicals in very low concentration in the water, and use an integrated sampler to measure that? And you all know, I think, where I'm going with this. And the answer is sure there is. And it's this: organisms, right? The organisms are integrative samplers that live in that environment. So whether you're talking about a crayfish or an insect or a clam or a juvenile salmonid or a smallmouth bass or a bald eagle or a mink, any of those animals, if they're in that environment long enough, they're going to accumulate those chemicals. And as you move up the food chain, they're going to biomagnify those chemicals. So these are great organisms to be used to assess concentration of things like mercury in the environment. All right, the EPA report in 2009 fully understood this. And they came out with again, this quote from the report: environmental indicators are animal species living in the Columbia River Basin or dependent on food from the Columbia River Basin that can be used to help citizens and decision makers better understand the health of the environment. So they were right on in what they were saying of let's did I just lose the slides? I apologize. Okay, are you guys still seeing my slides? Lori, are you guys still seeing my slides? Okay. Bear with me here 1 second. I apologize. Let's see. Because I'm not seeing my own slides, and that always makes, there we go. I apologize. I got overly animated and I touched the wrong button and I lost my slides for a second. I apologize. Okay, they're back. All right, so the Columbia River Basin, the EPA knows that we can use these animals to assess the environment. Okay, great. And again, one of the values of this is that relative to looking at the water, POPs, these persistent organic pollutants, and mercury both biomagnify up the food chain. So if we're looking at things like aquatic insects or insect-eating fish, and you would put crayfish about at that place where the insect-eating fish are on this slide, it's actually much easier to measure the concentration of mercury in the animal than it would be in the water. So this is really a really valuable thing. Now, there's a problem here, or a caveat. And if you go back to the Columbia River Basin State of the River report, these are the animals that the report is supporting having be used for environmental monitoring. Things like predatory birds, juvenile salmon, resident fish, smallmouth bass, semi-aquatic mammals, things like mink. They also have in their list, they have one invertebrate, and the invertebrate is the Asian clam, which is a problem because the Asian clam is an invasive. It's not even native to the environment, but they were making the argument that this would be a valuable environmental sentinel species. This is a problem, and the problem is many of those animals on that previous slide are charismatic megafauna, things like bald eagles. As much as we want a safe environment, you're going to be hard pressed to find anyone who would make the argument that shooting a majestic bald eagle out of the sky in order to determine its mercury concentration was valuable. So these animals aren't approachable. They're not approachable from a scientific perspective. They're certainly not approachable from a community perspective. What about these? People have been using invertebrates to assess stream quality for the last 50 years. So these animals, including crayfish, are readily available to communities to help assess the environment. All right, so what can Columbia River crayfish teach us? Well, first of all, crayfish, just like many of their sibling invertebrates, right, their sister invertebrates, crayfish accumulate non-metabolizable compounds, these persistent organic pollutants, in a very linear manner, in a consistent manner. Therefore, they're incredibly useful for monitoring things like DDT, PBDEs, PCBs, and of course, mercury. So the first thing right off the bat, you got the right animal for the right job. So that's number one. All right, let me talk a little bit about the Crayfish Mercury Project and the objective of the Crayfish Mercury Project. And our long-term goal, as it says here, is to be able to take data from any species of crayfish collected across the entire basin, standardize its mercury concentration, and what I mean by that is standardize for the species, so there may be species to species differences, standardize for differences in things like sex or mass or reproductive status, standardize all of that so you can boil the data down to a single number for each site, and then quantitatively add that information to a basin-wide map. And the reason that we would want to do that is we would want to effectively generate geospatial layers, right? So the bottom layer here is the watershed geography. We could then overlay on that watershed geography the concentration of mercury in crayfish. We could overlay on top of that finfish mercury, salmonid or resident finfish mercury concentration on top of that. And this could start to be able to give us a geospatial evaluation of what the basin looks like. So that's really where we want to go. The problem is that, as we all know, the Columbia River Basin is pretty big. So if I were to take my graduate student who's listening to this conversation as we speak, if I was to take him and maybe a few undergraduates or some other people, put them in vans, trucks, and have them ride around the basin collecting crayfish across the entire basin, it's going to cost me a lot of money. It's going to cost me a lot of time. It's going to cost a lot of analytical expenses. And in the worst case scenario, none of you are ever going to know about it, right? Because we're going to be doing that in our ivory tower at the University of Idaho, and you're never even going to know it's happened. And that doesn't do us any good. It doesn't do you any good. It doesn't do anybody any good. So, and we can do better than that. All right. So we want communities to help. We want communities to help and get involved in this process. So the first question we have to ask is is this really even possible? Well, the value of using you, right, using community members is, my student has to get in a truck and drive a day to get to Portland. You live there. For some of you, I noticed you live there, right? So you can go to a stream in 20 minutes. Takes my student 6 hours. So there's a widespread geography of that can be covered. We can do rapid data accumulation, and if we wanted to, we could have scores of citizen science groups collect simultaneously. So you're all collecting animals on the same day. We could do that. We've done that historically for other projects. That's possible. So there's this capacity for rapid data accumulation. We can mobilize large number of individuals at one time, as I just said. And it also gives you a piece of empowerment, right? Like, I'm not just thinking, I'm not just sitting home worrying about 6 PBT cube quinone or mercury or PBDEs. I'm actually part of the solution. I can actually go out there with my kids, with my Boy Scout group, with my church group, whatever else, and participate in this. And that is completely possible. All right. The gold standard for doing this is the Christmas bird count. And the Christmas bird count has been going on, started in 1900, so it's been going on for 124 years. It's always conducted on Christmas Day, which is interesting because Christmas Day is very close to the winter solstice. Therefore, what they have been able to do over time is collect now stay with me on this, collect the most northern extension of southern fish on the shortest day, or excuse me, not fish, the northern extension of southern birds on the shortest day of the year, right? Effectively the solstice. That's really important because what you can see in this data very, very clearly is over the last 125 years, that northern extension of southern birds is moving further and further and further and further north as the globe warms. So these birds are being huge environmental sentinels for climate change. And the data are absolutely compelling. All right. Well, what about water and environmental and chemicals and my experience with it? And so kind of zooming up from 1900 up until the last 10 years, my colleague and I, Dr. Heiko Schunfuss out of St. Cloud State, put out 2 papers in 2011. And in those papers, we argued that taking that approach that was being done by the Christmas bird count and applying it to environmental contamination in water bodies was really valuable. Back then, the reason I'm telling you this is back then in 2011, which really wasn't that long ago, the question was really, is citizen science viable or not? And we were making arguments in these papers that it was. Fast forward to April 2024, and the US EPA has designated April as National Citizen Science Month. And what that has done is it's no longer a question of whether citizen science is viable or not viable. It's a question of the US EPA, as well as other federal and state agencies, have absolutely jumped on board and are total supporters of using animals like crayfish and using communities to aggregate those animals. They're absolutely on board with that. There's money out there, like through EPA Region 10, to support that. And it is really a burgeoning field within the field of environmental monitoring. And as Lori said earlier, I'm extremely bullish on this and really, really excited about it because we've been kind of banging this drum, us and others as well, of course, but we've been banging this drum now for almost 15 years, and it's nice to see this finally come to fruition. All right. But really, is this possible? Okay, theoretically, is it possible? It's possible, but really, is it possible? Yeah, it really is possible. And I wanted to show you some ongoing work that's going on using these approaches. And it dovetails in both human health and environmental health. For example, there is a tick encounter group, which is aggregating data, is actually aggregating tick bodies that people acquire, pull off their own skin, send to them, and then they look for things like tick-borne diseases. And they assess the geospatial distribution of tick-borne diseases on a national level through this tick encounter program. There's a very similar program using there's a very similar citizen science program using invasive mosquitoes, doing a very similar thing. Again, looking at the mosquitoes as vectors of arthropod-borne diseases. Maybe more close to home relative to environmental health, there's on the coasts, there's the Mussel Watch. And what the Mussel Watch does is use blue mussels as environmental sentinels for contamination. And there is also, much more recently, the Dragonfly Mercury Project, which is a project out of the USGS. And they are also looking at dragonfly nymph, before they have wings, the dragonfly nymphs, and assessing them for mercury concentration at national parks nationwide. So this is real. I mean, this is 21st century science that's going on, and we're trying to match that with the Crayfish Mercury Project within the Columbia River Basin. So the community is important, and we've done a lot of work with community members. Here's just some of my graduate students and some of the community members out in the field collecting these animals. The really pretty signal crayfish, the really pretty crayfish with the white claws, the white marked claws is a signal crayfish, one of our natives. So this guy's not an invasive; he's a native to our waters here. And the people go out and they have an absolute ball catching these guys and helping us out. Thus far, here's a map of the sites that we have collected at thus far. This is the basin, and it doesn't have the states. So for me to orient you a little bit, the mass of red circles at the top is effectively eastern Washington and northern Idaho into northern Montana. The collection of dots in the lower right is the Boise River, and the collection of dots in the lower right is Boise, lower left is John Day. But you'll notice we haven't collected in the Willamette. We haven't collected in Hood River. We haven't collected further west, which is part of the reason that I'm talking to you today, because we're going to be moving in that direction. Literally, our data collection is going to be moving in that direction soon, meaning this season. And there would certainly be opportunities for you to be involved with us if you so choose. All right. These are the data that we've collected thus far. So these are total mercury concentrations in the adductor muscle, the big tail muscle of the crayfish, the muscle that if you live in Louisiana, you squeeze that out and you eat it. Well, I guess people in the Columbia River Basin eat crayfish too, so I shouldn't just be biased to Louisiana. But it's the mercury concentration in that adductor muscle. And I'm not going to spend a great deal of time on this today relative to the data analysis, because I have a couple of other places I want to go. But you can see that there are some sites that on the far left on this slide, Peoples Park, Harvard Park, that have really, really low concentrations of total mercury in these tails. And then there are other places like Walnut Creek, which are substantially higher. Now, importantly, none of these animals are unsafe to eat. So if you happen to be camping near Walnut Creek and you had caught a number of these animals and ate them, you're not going to get mercury poisoning or anything like that, happily. But there is variation within the environment. All right. So the second thing that crayfish can teach us is that crowdsourced crayfish collection by community members is a really, really powerful tool. And we couldn't have done the work that we've done so far without your help. So we really appreciate it. And we look forward to working with communities in the future as well to help us. All right. I wanted to take a few minutes, before I stop and open this up for questions. I wanted to take a couple of minutes, and I just wanted to talk about some burgeoning issues that are coming up. And the first one is PFAS. So I wanted to talk about PFAS and its relationship to everything that I've just been talking about. So PFAS, I'm going to go back for a second. These chemicals are these persistent and mobile organic chemicals, meaning that we can also call them as a group these PMOCs. All right, so these PMOCs, as I just said, are water soluble, persistent chemicals, and their biotransformation is minimal. So forever chemicals, right, like PFAS, they're not going to break down in the environment, but they're water soluble. So they're really, really different than mercury or PBDEs or PCBs, which are lipid soluble. These chemicals are very different. So their fate in the environment, their transport in their environment, where you're going to find them in the environment, really, really different than what I was talking about earlier. Now, relative to PFAS, and why I put this in here today, is that some of you may be aware that the EPA just came out on Wednesday with its final PFAS national primary drinking water regulations. And I just picked one of the PFAS molecules to talk about a little bit, PFOA here, this perfluorooctanoic acid. And its regulatory standard for drinking water is 4 nanograms per liter. And you can see I drew it out underneath. That's a really low concentration. That concentration, and if you remember my very first slides, that concentration is low enough that we're at the limits of analytic capability. We can measure it, but that's about as low as we can go. 4 nanograms per liter is really down there analytically, really hard to measure. All right, so this is what we environmental scientists, this is what we're dealing with from now on. This is kind of the gold standard from EPA of the toggle point at which these chemicals are considered to be important relative to drinking water. What about PFAS? What about crayfish? Can they be used as environmental monitors relative to PFAS? Is that even possible? Well, PFAS, first of all, like many of the chemicals I talked about before, these PFAS, as you're probably all very well aware, is not a single chemical, but there are many congeners, sister chemicals to that one chemical. So there's a suite of different chemicals, which makes it really hard because you don't want to follow them all. You just want to follow the usual suspect, right? But the question may be, who is the usual suspect that you want to follow? These chemicals, as EPA just testified in the previous slide, are bioactive at extremely low levels. Their solubility, the fact that they solubilize or mobilize in water, favors their mobility. They're not going to aggregate within the tissues of animals like mercury does or like PCBs do. They're more labile than that. They tend to be more mobile. Is that going to be a problem? Also, though, the chemical is mobile, as we all know, but it doesn't degrade. It's one of those forever chemicals, so it's going to be around, moving around in the environment without degrading. Well, crayfish, can they be used? Well, here's a study that I just that I pulled from the literature. This is a recent study in the journal Chemosphere, and I just wanted to point this out to you all. And that's the fact that what this is showing is two specific PFAS molecules, PFOA and PFAS, and it's showing the pooled BOF on the y-axis to the left. That pooled BAF is bioaccumulation factor. So how much does the molecule accumulate in organisms? And as you can see here, what this slide is telling you is, yes, you could use invertebrates such as mayflies, water pennies, caddisflies, stoneflies, and crayfish. You could use them as environmental monitors to assess PFAS. And this is one of the things we're looking at in the future, is using these animals to do just that. So a third thing that the Columbia River crayfish can teach us is that, yeah, they're ready, willing, and able to be part of a fate and transport program for PFAS. They can accumulate the chemical, it can be measured in their system, and they're ready to go. So we could actually do that. The final thing that I wanted to talk about is, and again, another, I'm going to use the term chemical, although it's not really accurate. Another chemical that's burgeoning on the scene that we in the Columbia River Basin are going to be dealing with for the next few decades are microplastics. And the question here, right, is are microplastics the next emerging contaminant? And if they are, how do you regulate an emerging contaminant that's not a chemical, right? Effectively, if you think about, like in this photo or this diagram on the left, if you think about a water bottle, and if you think about this water bottle lying on a beach somewhere in the Columbia River Basin, and it's degrading over time. Now, degrading, I just mean it's simply breaking down into smaller and smaller pieces. It's the chemicals themselves are effectively POPs. They're persistent organic pollutants. They're not going to, the chemical itself is very resistant to breakdown. But those chemicals, those bottles or the plastic will wind up in progressively smaller and smaller and smaller particles. And as those chemicals get smaller, they start to leave the realm of macromolecules or particles, and they start to enter the realm of chemicals. And it's mind-boggling to think about it, but there is a point in size where they kind of flip over from being really tiny particles to really enormous macromolecules. And then they start to behave very, very differently. And again, I pulled another paper, Dent et al. From Ecology and Evolution, and I just wanted to show you, these are micrographs of signal crayfish muscle, the same muscle that we use for mercury analysis. And here are suspected microplastics, microfibers that have embedded themselves from the environment into the muscle tissue of this animal. It's really astounding. It's astounding that it can be identified, but again, it's astounding that these microparticles are maintaining their integrity at very, very small sizes, and are just that the particle is just getting smaller and smaller and smaller, but it's not degrading in the way that something like a steroid or something else would degrade. Really incredible. But again, what this shows is, yeah, you can use signal crayfish to environmentally monitor for microplastics. The last thing that I wanted to talk about is this relationship when these microplastics become really, really small. There becomes a relationship where effectively, if you think about a chunk of again, a plastic bottle, which is that big cloud over on the far left, as that as that plastic breaks up into smaller and smaller pieces, the surface area of those particles, the particles themselves get smaller, but the aggregated surface area gets larger and larger and larger and larger, right? And you can think about these things as like little tiny crackers. Now, bear with me here for a second, right? So you have these very small cookies or very small crackers. Now, applied to those tiny pieces of plastic, what you very well may find, because of the chemical affinity, is that things like POPs, PFAS, other chemicals will aggregate and bind to those tiny particles. So now you have a tiny cracker completely embedded in peanut butter. And it may be that the peanut butter itself is the toxic element riding on the tiny cracker. This is something that trust me, we'll be talking about this again in 10 years, but this is kind of on the forefront or on the edge of where environmental monitoring is going. And again, if crayfish are able to aggregate these tiny particles in their tissues, which has already been shown by that previous study that they can, we'll be able to address this using those animals. So what can Columbia River crayfish teach us? Finally, crayfish can help us understand the environmental fate and transport of microplastics and the contaminants that those microplastics may carry. They can help us understand PFAS. They can help us understand mercury. They can help us understand other persistent organic pollutants. We have in this animal and his sister invertebrates a gold mine of information that we can assess. And all we need is the will and frankly the money to do it. But the operational capability is already there. So before I stop and take questions, I'd like to thank the team that we work with. And by that, I mean the citizens. We've worked with K-12 kids, we've worked with professional organizations, we've worked with tribal communities throughout the basin. We've been really humbled by the interest and the enthusiasm of the community relative to helping us, and we look forward to that continuing, frankly. And with that, I appreciate your attention. For some of you, maybe I should apologize post hoc, but this may not have been where you thought this presentation was going to go, but I wanted to really focus on the community aspects of this and the capability that invertebrates have for interfacing with the community and how it's working today and how it also might work in the future. Two other things. First of all, as Lori said earlier, shameless promotion. I did write this book called Modern Poisons. This book we're going to raffle off one of those to one of you today. The whole point of Modern Poisons was that I'm really passionate about taking the information, like what I talked to you about today, and providing it to individuals, K-12 kids, whoever else, in the language that they can understand. And that's what this book absolutely tried to do. It's a paperback, it's short, it's inexpensive, yada, yada, yada. I'm not trying to get you to buy the book, but I'm trying to explain to you the audience that I wrote the book for. This is not a 400-page textbook written for graduate students at the University of Idaho. It's written for community members so that they can get a handle on these contaminants and what these contaminants are doing. Finally, if anything that I said today is of interest to you, get in touch with myself, akolak@uidaho.edu, or my graduate student Tate, T. Libanow at uidaho.edu, and we'd love to talk to you. So with that, Lori, I'll turn it back over to you.
Thank you so much. That was great. Yeah, and we will be raffling off one of those books. We'll also be raffling off some Columbia Riverkeeper swag. So if you were one of the winners, we'll be reaching out to you after the webinar. And I just, so I want to remind everyone, if you have questions, we do have a few minutes. You can pop those into the Q&A. And while folks are doing that, I just want to say, if this webinar leaves you wondering how you can support this work further, I definitely want to encourage folks to continue learning more about these issues. We will be sharing in a follow-up email to everyone, we will be sharing a video recording of this webinar, so please share it with friends, folks, whoever you think might be interested in this. We'll also be sharing Columbia Riverkeeper's mercury fact sheet if you want to get a little more understanding about the kind of issues around mercury on the Columbia River. And then because these issues were talked about, I just wanted to mention too, we also have webinars that we've done on both microplastics and PFAS in the past year and a half. And those are all on our website and available. So please, go check those out if you want to learn more about those issues. Yeah, so with that, I will jump into the Q&A. And I see the first question is, an anonymous member said, I am interested in knowing if Columbia River crayfish are safe to eat, how much and how often?
The short answer is, the short answer is I can only speak to that question relative to mercury, which I'm sure that's what the questioner was asking about specifically. And the answer is yes. As I said in kind of in the middle of the talk, we have yet to find an individual crayfish that exceeds the EPA fish advisory standards for mercury. So yes, you can eat them. And how many and how often at this point, unless you're a pregnant woman, if you're a pregnant woman, then the whole game changes because of the fact that mercury bioaccumulates, and you just need to be uber careful when you're in that status. But if you're an old male like me, you can pretty much eat as many of them as you want, whenever you want, and you're going to be fine. Gloria, you're muted.
Sorry about that. Thank you. I'm going to combine a couple of questions because there's some similarity. But so one question is about the sampling. So do you extract the crayfish from the watery or from the waterway? Are you are people collecting them and then sending them in? Or are folks collecting, the sample and doing some analysis on their own? Because that ties into another question somebody had about, are there differences in instrumentation or the way, is there some bias in the way it's measured in one location versus another?
To date, so let me address that last question first, right? A crayfish, as we all know, a crayfish has an exoskeleton. So effectively what that means is that, and if you think about it relative to the integrative samplers I was talking about earlier, the sampler, if you will, which is the muscle tissue, is inside of a relatively impregnable box. So you can pick up the crayfish and you're not going to contaminate, the mercury on your fingers isn't going to contaminate the tissue level inside the carapace. So from that perspective, collection is fairly easy in that you don't have to use specific tools and things like that. Now, if we were to get into PFAS or microplastics, then the game changes because cross-contamination would become a lot more likely, and that would be a little bit more problematic. So there's that point. Where we would like to go with this study is we would like to have a sliding scale. And what I mean by that is we'd like to have a sliding scale where if you were a member of an organization like Columbia Riverkeeper, and you said, okay, we want to look at Hood River, and we want to assess crayfish within Hood River. we would say, okay, how, I'm trying to think of the word to use. How involved in this do you want to get? And I don't mean this in any negative way, but like, how serious are you, right? Because if you were to say, well, if we want your graduate student to drive down, have a engagement with us, send us out into the field, we collect the animals, we bring them back to your graduate student, and he takes them home, we can do that. But if you were more like, no, we really want to get into this and we really want to be doing it, then that can also happen. And we can talk about how that could potentially happen down the road. Most of our interactions to date have been those kind of level one of, we'd like to have your student or you or whoever else come to us, come down in a van. We do the thing in a day. We have in a way, like a bio blitz in a day. We collect 50, 60, 70 animals from 10 different locations, whatever, and then we send you along your way. But again, if an organization or a community wanted to be more involved than that, we can certainly work out those details.
Awesome. Thank you. And there are definitely some more questions. I do want to be respectful of everybody's time. There are quite a few questions about how to get involved and how many samples are needed, and when can I do this in Portland, which is awesome. Love the enthusiasm. And in the interest of time, I just want to say to folks, please reach out to Riverkeeper, reach out to Dr. Kolak, after the webinar, to kind of follow up on some of that if you're really excited about getting involved and wanting to engage, at a deeper level with this. That's awesome. We fully support it. So with that, I think, we might need to kind of continue this conversation offline, and folks can, reach out to us, and, we can answer questions as best we can that way. But I do want to say thank you so much to everybody who joined the webinar today and took the time, and a huge thank you to Dr. Kolak for taking the time today, sharing his expertise. And yeah, we just really appreciate everybody being here. There's a lot of great conversation happening in the chat too, but please, let's continue this and reach out to folks online. So with that, I want to say thank you so much, and I hope everyone has a great day.